Have you ever wondered how DNA replicates? DNA, this intricate double-stranded molecule, must first unwind into two single strands to kickstart the replication process. Imagine the strands like a twisted ladder, where the rungs need to separate to form two new ladders. This unwinding is initiated at specific regions which are rich in adenine and thymine. These regions are known as the origin of replication, or ORI for short. It's like the starting line in a race where the replication process begins. These origins serve as targets for initiator proteins, which call in reinforcements in the form of more proteins. These helpers assist with the replication process by forming a replication fork around the ORI. Picture this like a team gathering around the starting line, ready to begin the race. As the saying goes, every journey starts with a single step. And in the case of DNA replication, the formation of a replication fork exposing two single-stranded strands marks the beginning of initiation. So what happens after the DNA unwinds? Well, this is where our microscopic heroes, the enzymes, come into play. The enzyme DNA helicase takes the center stage. Acting like a little molecular motor, it unwinds the DNA from its origin of replication, exposing two strands in a Y-like structure. This structure is what we call the replication fork. Now, imagine pulling a tightly coiled spring apart. It would surely want to spring back into place, right? Similarly, the activity of helicase causes what we call topological stress to the unwound strand, forming what's known as supercoiled DNA. But nature always has a solution, and so does our DNA. The stress is relieved by another enzyme called topoisomerase, which uses negative supercoiling. So here's the fascinating bit. The replication fork is bidirectional. One strand is oriented to 3 to 5 direction, and the other strand is oriented to 5 to 3 direction, but the addition of nucleotide progress only in 5 to 3 direction. Now that we've gone through the process, let's summarize the key points about DNA replication. DNA replication kicks off with the unwinding of the double-stranded DNA molecule, an action facilitated by breaking the complementary base pairing. This unwinding process starts at specific coding regions, known as the origin of replication, or ORI for short. The ORI is targeted by initiator proteins which recruit other proteins to form a replication fork. The enzyme DNA helicase, part of the replication protein complex, takes the lead in unwinding the DNA from its ORI, exposing two strands in a Y-like structure, our replication fork. However, this activity causes topological stress, forming supercoiled DNA, a stress that's relieved by topoisomerase through negative supercoiling. The replication fork is bidirectional, with one strand oriented 3 to 5 and the other 5 to 3, but nucleotide addition progresses only in the 5 to 3 direction. Understanding these steps gives us a deeper appreciation for the intricate process that is DNA replication.